Welcome again to Physiology Lectures. Now we start into a new chapter, Chapter 17, Part 1. This is going to cover a lot of breathing mechanics, how we get air in and out and set us up for gas exchange, which we'll talk about in Chapter 18. So in this chapter, we're going to cover sort of three main sections. We're going to do a little bit of anatomy, especially at the micro level of the respiratory system. We're going to put the phys and physiology yet again, talking about gas laws, trying to help make these laws accessible to you. I realize that gas laws and physics often feel relatively abstract. We're going to try to make it a little more real for you. And then lastly, the mechanics and the physics of ventilation, how we get air in, get air out, and potential obstructions or impediments therein. A few respiratory system functions, some of which we have known about probably all of our lives, and then some which may be a slight twist on what you've thought about when it comes to breathing and gas exchange. So yes, we are breathing, we are moving gases between the atmosphere and the blood. Breathing fundamentally is sustaining diffusion gradients. By keeping oxygen levels high, we maintain a unidirectional flow of oxygen. By maintaining CO2 levels low in the alveolar space relative to the blood, we continue to move carbon dioxide out. And make no mistake about it, carbon dioxide is just as critical to life as oxygen. We certainly give oxygen a lot of credit, but we shouldn't be forgetting about carbon dioxide because carbon dioxide is the backbone of regulating body pH. If you study body pH disorders, typically they'll classify it as either respiratory or metabolic acidosis or alkalosis. Well, respiratory is a big factor. Gas exchange is a major regulator, moment to moment, of pH within the body. If you stop breathing, you begin to cause acidosis in the blood, and if you hyperventilate, you begin to cause alkalosis. There's no secret to why you start hiccuping if you laugh really, really hard, because what's happening is your body is sensing impending alkalosis and interrupts the breathing cycle in order to slow it down. We don't think much about the protection side of our respiratory system, but it is a relatively thin barrier between the external environment and our internal environment, and it is a narrow spot where pathogens can take advantage. We don't need to look any further than what's going on with COVID-19. But it's protection from not only pathogens, but also irritating substances. And irritating substances we would define as things that drive inflammation or potentially cause injury. So we need to make sure that we're protecting from those things just as much as pathogens in many cases. And then lastly, I always like to incorporate psychology and complex behavior as part of the physiologic process. It really is a shame that we don't incorporate our central nervous systems as part of fundamental physiology, but vocalization is very much a physiologically relevant behavior. If for some reason you need help, you need assistance, you would vocalize that. If you are trying to achieve some goal, vocalization is often very, very critical to that homeostatic end. So vocalization is just as important as any of these other things for obtaining physiologic goals and sustaining homeostasis. So the act of respiration, which is the act of moving gases, is fundamentally a bulk flow system. We are changing pressures in order to achieve gas movement. So we increase pressure inside the chest, the air moves out of the chest back into the environment. We decrease pressure inside the chest, that causes the environment to move into the alveolar spaces. And it's driven by muscular pumps. The elastic recoil on expiration is also used, but we can also use muscular pumps for both inspiration and expiration. In terms of movement of air, we also have to take resistance into account. Under normal circumstances, resistance is relatively low, but if we have inflammation or obstruction, the radius of these tubes that allow for gases to move into the lungs or out of the lungs can quickly impede the ability for gas movement to occur, either by increasing the resistance or outright occlusion. The three major components of the respiratory system include the conducting system, otherwise known as airways. This includes areas up in your nose and mouth, through your larynx and trachea and so forth, down into the lungs. And a significant fraction of lung volume is comprised of airways as well. The exchange epithelia, the structure known as an alveolus in singular or alveoli in plural. Not just epithelium, but also other tissues involved in that as well. Not going to talk a ton about these except for the idea of movement of air, but certainly bones and muscles and connective tissue. From a diagnostic anatomical standpoint, and I say diagnostic anatomical because whether we're in the upper respiratory system or the lower respiratory system may mean the type of intervention that occurs in terms of alleviating issues in the respiratory system. So the upper respiratory system is pretty much defined by sort of that fold you can feel at the top of your Adam's apple. Right in there in that spot is basically where the upper respiratory system ends. But it includes your nasal cavity, tongue, larynx, and upper trachea, if you will. By the way, some of the initial tests for COVID-19 require swabs in this area of the upper airway system, though the newer tests seem to not require such a deep penetration into that system. But the gold standard right now requires that deep swabbing in this area. The lower respiratory system, on the other hand, starts there at about your Adam's apple and works down into the lungs themselves. And again, the anatomical significance of these two is really about intervention as much as anything. Turns out pediatric cases often involve the upper airway systems that are much more significant in peds than they are in adults, just because their airways are so much smaller. And we have two lungs, generally speaking, the left lung having 2 lobes, the right lung having 3. Just a little bit of emergency medicine trivia for you. If you inhale or otherwise aspirate an object, most of the time it'll deposit in the right lung by virtue of the asymmetry of the 2 major bronchi right here. This one's a little more vertical, and odds are it'll be over on the right side. Just again, a little bit of trivia for you. The lungs in the pleural cavity are a 2-sac system. You have an air-filled balloon, and then you have this fluid-filled balloon around the outside, known as the pleural membrane. So it's a 3-layer system, not unlike the pericardium, that secretes a small amount of fluid. And the point of that fluid is to allow for rapid and significant changes in volume without too much irritating friction against the chest wall. Because we can significantly change the volume. You can take in 3 or more liters of extra air above and beyond your normal inhalation. And that's a significant change in volume. And if left unchecked, left unlubricated, would cause significant irritation. Many of the folks that are suffering from COVID-19 right now are probably also suffering from mild pleurisy, where it's exceptionally painful to breathe in addition to difficulty breathing. And that's due to irritation of the chest wall and the lungs during this process. The other point of having this fluid is to help retain negative pressure inside the pleural cavity relative to the outside atmosphere, usually 2 or 3 millimeters of mercury lower than normal atmospheric pressure. Why is that? We want to keep this lung maximally in contact with the chest wall. We don't want to develop a gap in there, or we won't be able to change the volume of this lung easily or at all. We want to be able to vary pressure inside here, and to do that we have to change volume. And to change volume, we have to have positive contact against the chest wall, or it's not going to work. We'll talk about pneumothorax later on in this chapter. We do have a circulation system that's independent from the regular systemic vascular circulation. It's a lower pressure system, little less than half of the pressure than what we see in the systemic circulation, coming off of the right ventricle. So we have the pulmonary arteries, goes to the lungs where it bifurcates down to a capillary level, returns via the pulmonary veins and back to the left atrium. Again, a lower pressure system. The wall of the myocardium here is much thinner and very much less instances of heart failure. In fact, if anything, if the left side starts failing, the left side starts causing a fluid back up into the lungs. And one of the most common signs of left side failure is pulmonary edema, buildup of fluid here because the right side is outpacing the left side. When we talk about respiration, we can actually use respiration for more than one thing when it comes to physiology. So in most cases moving forward, if I remember, I'm going to use the word for the exchange here, the first level of exchange, as ventilation rather than respiration. Sometimes people will call this respiration, but we also have cellular respiration. So in order to sort of differentiate it, I'll be trying to call this movement, this exchange one between the atmosphere and the alveoli, as ventilation. That's exchange one. The second level of exchange is here in the alveolus with the blood plasma and the red blood cells. Remember how I say it in that order: blood plasma first, and then the red blood cells second. The blood plasma always comes in between the cells and the exchange between the alveolus and the blood. And then the third thing, so there's exchange one, there's exchange two, and then our third exchange is down here at the level of the tissues. And again, it goes from cell to plasma to extracellular fluid to plasma to cells. And that's exchange three. And then we have the manufacturing of ATP that we refer to as respiration as well. And so the circulation here is just as important as the ventilation here in terms of sustaining gradients. We're going to talk about VQ ratio a little bit later, the V standing for ventilation and the Q standing for circulation. And so we start at the larynx, move down the trachea, and we bifurcate and bifurcate again and bifurcate again, bifurcate again. So starting with one trachea, we end up with 80 million bronchioles right before we go into the alveoli, and there are at least several alveoli for every bronchiole. So we have an extraordinary level of detailed architecture here. It's created by a process in developmental biology called branching morphogenesis. Very important for kidneys and other blood vessels in the body, but it's super duper cool because you have literally at least 22 bifurcations all the way before you get to the alveoli. So extraordinary increase in surface area to maximize gas exchange. So we have it right here, and by the time we've reached the alveoli here, we now have a surface area somewhere around 1 million square centimeters, which is roughly the size of a full tennis court. Pretty impressive, the amount of surface area we can achieve. But it also does create some physiologic, technical issues that we have to overcome, one of which is surface tension. The smaller the radius down here, the higher the surface tension, and that can lead to issues with fluid accumulation. And we'll talk about that in this chapter. Just for fun, the most complex river system in the world by far is the Amazon watershed. It extends completely across the continent of South America, which is roughly, if this is, let's say, Washington, D.C., over here is like Denver. It's enormous down there. I had no idea until I flew over it. In any case, the level of complexity in this river, you have 12 bifurcations when you go to the headwaters, whereas in the lungs you have 24. So the architecture is roughly twice as complicated as the most complex river system in the world. Pretty neat stuff. Because we are dealing with a fundamentally diffusional event at the level of the alveolus, those alveoli are very, very delicate, very, very thin membrane. And as I mentioned earlier, one of the roles of the respiratory system, the pulmonary system, is to protect and prevent pathogens and irritating material from getting into the body, and that begins at the alveolus. We need to condition the air. Conditioning air, like an air conditioner, what we're doing is we're changing the temperature, changing the humidity of the air. Not necessarily lowering the temperature, but changing it to a more suitable temperature and humidity, and also filtering out potential irritants. So we're going to warm it to body temperature. Again, remember diffusion is proportional to temperature. So we want to warm it in order to increase the rate of diffusion once it gets to the alveolus or close to body temperature. We want to humidify. Why? Because those membranes are delicate and they do depend on fluid in order to retain their permeability. If we start to desiccate those membranes, what happens is those phospholipids start getting clustered together in a crystalline form. That's why it gets very, very brittle. If you've ever let something desiccate, okay, it gets relatively brittle. And so we keep the water vapor there in order to ensure the membrane fluidity on the interior surface of the alveoli. We're going to filter out foreign material, filtering out those irritating substances where possible. Unfortunately, if you inhale a whole boatload of it, and it doesn't matter if it's coming from a tobacco cigarette or a marijuana cigarette, you're still inhaling burning vegetable matter, okay? You're still inhaling lung irritants. Don't let anybody else tell you otherwise. It's still smoke, okay? And it's still going to cause irritation and injury. So we're going to try to filter that out as best we can. Or in a more natural situation, like for instance, if we see dust in the air, like during a relatively strong thunderstorm, pollens and other types of air impurities, we want to get rid of those before we hit the alveoli so they don't irritate and otherwise impede alveolar function. Part of how we do this secretion and prevention of pathogens and so forth is through secretion, either through a submucosal gland like this or a goblet cell, which secretes a protein known as mucin. Mucus, the material that we associate with things like in our nose and so forth, is not a homogeneous substance. The stuff you have in your nose is actually quite full of dirt and contaminants and has been transported to that area in part due to secretion, but also in part due to ciliary function. And mucin can be very, very viscous, like in your nose, particularly if you're dehydrated or so forth, or it can be exceptionally liquid. Mucin protein is very, very hydrophilic. Very, very hydrophilic, which means it wants to trap water. And so depending on the ratio of mucin to other proteins, it can be exceptionally thin. It can be exceptionally almost like a light machine oil compared to the viscous stuff that we're normally associated with. So a combination of secretion and ciliary movement is what we do to condition the air and filter the air as it's moving down the conducting airways. And so secretion going from the interstitial fluid to a more external space. We're going to show you a very, very specific mechanism for how we get water from the interstitial fluid to the ciliary surface of the airway epithelium. And so here is a submucosal gland. This gland right here is one of the glands responsible for getting water to the surface of this epithelium. And the key here is the chloride anion, the Cl minus. This is the first time we've really talked about a physiologic role for Cl to get water onto the surface of this membrane. Super important, and that's the basis for something known as cystic fibrosis. So we're going to talk about this mechanism here, how we transport chloride to the exterior surface to facilitate water movement in order to humidify and liquefy the mucin that's already been secreted from the goblet cells. Here is just a simplified gland epithelial surface here. Here's the apical surface in the lumen of that gland. Let's walk through the steps for how we get chloride to that surface. Well, the first thing begins down here at step 1. Step 1, we have a transporter here, and you notice it's carrying more than one thing. It's carrying sodium, it's carrying chloride, and potassium. This is a SYM porter, S-Y-M, more than one substrate in one direction. It's not an antiporter. They're both going in the same direction. Now, I know you're going, well, potassium's high in here. Why would it move? Good question. That's why we would know that sodium is driving this, because sodium would be moving down its transport gradient. This is secondary active transport. Secondary active transport, because there is no ATP hydrolysis here. It's over here. Here's the ATP hydrolysis. We're pushing potassium in, sodium out while we're hydrolyzing ATP. So the ATP hydrolysis is once removed. It's over here instead of here. So this is secondary active transport. And so this NKCC, which stands for sodium potassium chloride carrier, okay, just use NKCC for your own sake, forces chloride and potassium in by using the gradient of sodium to bring it in. And so we get chloride moving into this intracellular space. And by the way, it generates a relatively high gradient of chloride here relative to most cells. I know you're normally saying, well, chloride would be low. Well, that's true in most cells, but not here. We're building a chloride gradient. So we build this chloride gradient to allow for diffusion, facilitated diffusion. Remember, this is a hydrophilic and it's charged. It's not going to want to move across the plasma membrane. Facilitated diffusion out of the cell onto the apical surface of this gland. This channel has a name. They actually named it before it was actually discovered. They knew it existed, but they hadn't located it definitively. And so they named it CFTR. And the CF stands for cystic fibrosis. They didn't know it was a chloride channel. They supposed that there was some sort of channel that was impeding water movement to the surface, and it turned out to be chloride. So cystic fibrosis, terminal something, I forget, translocator or something, I forget what it is. In any case, it's this channel that allows chloride to diffuse down its concentration gradient onto the apical surface. Well, that creates a negative anion gradient on the surface. Well, so there goes chloride. I'm a little behind on my animations here. Well, sodium will move paracellularly. The tight junction, which is not shown here, it's maybe right around here somewhere, will permit sodium to follow chloride. So as chloride moves to the apical surface, you get a negative charge. And so those positive sodiums, which are quite common in extracellular fluid, will now move down its concentration gradient paracellularly to the apical surface. Lastly, since we have all these ions going on out here, lots of ions here, this creates an osmotic gradient that then allows for water to then move paracellularly. So we build up the sodium and chloride. Now we move water down here and we humidify this apical surface. What I will tell you right away is that if there's any problem with this guy right here, you can't move chloride, you can't move sodium, and then you can't move water. And that has life or death consequences. Under normal circumstances, people who have the cystic fibrosis gene may be asymptomatic. That is to say, it is an autosomal recessive disorder. In order to have the typical forms of cystic fibrosis, you have to have two mutant forms of the CFTR, which means you inherit one from your mother and you inherit the other from your father. And I have seen this be a nutcracker for many, many marriages. When they find out their kids are now susceptible to cystic fibrosis, it will rip marriages apart because cystic fibrosis is such a tragic disease. I had two in-law relatives die of cystic fibrosis. They became my in-law relatives after their parents had divorced because they were both cystic fibrosis carriers. In any case, if you have a normal CFTR from one parent and a mutant from the other, you're a carrier, but you are phenotypically normal. But if you have two mutant CFTR genes, you now can't transport chloride to the surface, sodium doesn't get there, and water doesn't get there. And so now the mucus builds up and it starts to create a very, very severe form of COPD, chronic obstructive pulmonary disorder. In the past, before modern therapies came along, most cystic fibrosis patients died by the time they were 20. Now the lifespan is a little bit older, 30s, maybe 40s, but it's still a significant cause of premature death. And turns out cystic fibrosis is one of the leading candidates for gene therapy, because if you can at least introduce a normal CFTR, and you don't have to have tons of them, you'll have a relatively normal phenotype. But it's not just the lungs. The lungs are secretory for the conditioning side of it. It turns out that chloride plays a role in the secretion of other things. And so they not only have breathing problems, they also sometimes have digestive problems and sweating issues as well. But the saying is that the reason people will starve to death with cystic fibrosis is they just don't want to eat. Because when they eat, they start secreting, and when they start secreting, they have trouble breathing. One of the manifestations of chronic hypoxia is this disorder with the fingers, this manifestation, it's not really a disorder, called clubbing. You see the kind of dome-shaped clubbing on the tips of the fingers? That is a response to chronic hypoxia for reasons we don't really understand why. You get this sort of clubbing phenotype. Late, when they have particularly severe cases of it that don't respond to other forms of therapy, what you'll see is this sort of cushingoid morphology appearing, because what they're trying to do is bronchodilate the lungs using steroids. So they'll use cortisol and some other things, and hypercortisolism drives this sort of moon face type of morphology that's called a cushingoid appearance. Very, very tragic disorder. Wouldn't wish it on anybody, but it fundamentally comes back to a very, very simple molecular issue, which is that you can't move chloride. And so we're at 28 minutes now. I'm going to go ahead and stop at this point for Chapter 17, Part 1, and I will talk to you again when we resume with Chapter 17, Part 2. Thank you.